Hello and welcome to today's lecture on cell biology. Today, we will be exploring the fascinating topic of cell proliferation and regeneration. Cell proliferation refers to the process by which cells multiply and increase in number, while regeneration refers to the ability of certain cells to replace damaged or lost cells. Understanding these processes is crucial for medical professionals, as they play a vital role in growth, development, tissue maintenance, repair, aging, and disease progression. So, let's dive into the world of cell proliferation and regeneration. Cell proliferation is essential for the growth and maintenance of organisms. It involves cell division, tightly regulated to balance growth and death. Regeneration replaces damaged or lost cells. Understanding these processes is important for fields like regenerative medicine and can have wide-ranging applications. Cell division is vital for an organism's growth. It begins with a fertilized egg dividing multiple times during embryonic development, forming different cell types. Throughout life, cell division replaces old or damaged cells. This process must be carefully controlled to avoid issues like developmental defects and cancer. In addition to growth and development, cell proliferation is also crucial for the maintenance and repair of tissues. Throughout our lives, our bodies constantly face challenges such as injuries, infections, or normal wear and tear. In response to these challenges, cells undergo division and proliferation to replenish the damaged or lost cells. This process plays a vital role in wound healing and tissue regeneration. For example, in the lining of our intestines, the cells are continuously dividing to replace the old cells that are shed from the surface. Cell division occurs through a precisely regulated series of events called the cell cycle. The cell cycle consists of several distinct phases, including interphase, mitosis, and cytokinesis. Interphase is the longest phase, during which the cell grows and prepares for division. It can be further divided into three subphases, G1, S, and G2. After interphase, the cell enters mitosis, which involves the division of the cell's genetic material into two equal sets. Finally, cytokinesis occurs, where the cytoplasm divides, resulting in the formation of two daughter cells. To ensure the proper regulation of cell proliferation, various control mechanisms are in place. These mechanisms include cell cycle checkpoints, which act as surveillance points, ensuring that the cell has completed all necessary processes before proceeding to the next phase of the cell cycle. Regulating proteins, such as cyclins and cyclin-dependent kinases, play a crucial role in controlling the progression of the cell cycle. Any disruptions in these control mechanisms can result in uncontrolled cell growth and the development of diseases such as cancer. Multiple factors influence cell proliferation. Growth factors, which are signaling molecules, stimulate cell division and play essential roles in tissue growth and repair. Hormones, such as estrogen or testosterone, can also influence cell proliferation in specific tissues. Additionally, cell signaling pathways, including those involving receptors on the cell surface, can transmit signals that regulate cell proliferation. Understanding how these factors interact and influence cell proliferation is crucial for developing potential therapeutic interventions for various diseases. Cell proliferation is regulated in a tissue-specific manner to ensure proper development and maintenance. Stem cells, which have the remarkable ability to differentiate into various cell types, play a significant role in tissue homeostasis and repair. Tissue-specific factors, including growth factors and extracellular matrix components, can also influence cell proliferation rates in specific tissues. Studying these mechanisms can provide insights into tissue regeneration and the development of targeted therapies for tissue-related diseases. Cell senescence refers to the loss of a cell's ability to divide and proliferate. As cells age, they undergo senescence, leading to a decline in tissue regeneration and repair capacity. This process contributes to the aging of tissues and the onset of age-related diseases. Understanding the mechanisms underlying cell senescence can help us develop strategies to delay aging and potentially prevent or treat age-related diseases. Uncontrolled cell proliferation lies at the heart of cancer development and progression. Cancer cells divide rapidly and uncontrollably, eventually forming tumors. Numerous factors, such as genetic mutations, disruptions in cell cycle regulation, and dysregulated signaling pathways, contribute to the development and progression of cancer. Understanding the mechanisms driving abnormal cell proliferation in cancer can lead to the development of novel therapeutic targets and treatment strategies. Apoptosis, or programmed cell death, is a critical mechanism for maintaining the balance between cell proliferation and cell death. During apoptosis, cells undergo a precisely regulated process of self-destruction. This process helps eliminate damaged, infected, or unwanted cells, preventing their uncontrolled proliferation. Imbalances in apoptosis can have significant implications, either leading to excessive cell death or prolonged cell survival, both of which can contribute to various diseases, including cancer. The cell cycle is tightly regulated at the molecular level by the activity of specific proteins called cyclins and cyclin-dependent kinases, CDKs. Cyclins and CDKs work together to promote the progression of the cell cycle by phosphorylating target proteins involved in cell cycle control. Different cyclin-CDK complexes are active at different points in the cell cycle, ensuring the precise temporal control of cell division. Dysregulation of cyclins and CDKs can lead to cell cycle defects and contribute to various diseases, including cancer. In addition to genetic factors, epigenetic regulation plays a crucial role in controlling cell proliferation. Epigenetics refers to heritable changes in gene expression that do not involve alterations in the DNA sequence. DNA methylation and histone modifications are two key epigenetic mechanisms that can regulate gene expression and control cell proliferation. Dysregulation of these epigenetic mechanisms can lead to abnormal cell proliferation and contribute to disease development. In conclusion, cell proliferation and regeneration are complex processes that are essential for growth, development, tissue maintenance, aging, and disease progression. Understanding these mechanisms is crucial for biology and developing treatments. We have explored various aspects and their role in cancer, aging, and repair. Continuing to study this field will lead to new insights and strategies. Thank you for joining me today.